Professor Davis here, and we are now going to dive into the immune system specifically and disease. Now, the part of disease that we're going to talk about are going to be mostly viruses because we already had a chapter where we talked about bacteria and we talked about diseases there. We also did protozoa and their diseases and fungus and theirs. And so this one's going to focus more on the virus structure. Then the second part of these notes is going to look at the vertebrate immune system. All right, so we're going to dive into kind of two separate parts in this set of notes. So when we're looking at viruses, viruses are considered non-cellular, and this is because they do not contain an actual cell. Okay, so they are considered non-living because of this. So they are more of what we would call a particle. So they're not in any of the domains. So they're not in archaea, eukarya, bacteria, and they don't have kingdoms either. Okay, so with taxonomy, they're a lot different. We also see that they're a lot smaller than bacteria. We see that they're approximately 0.2 to 2 micrometers, which is really small. And this is unique because this is what allows viruses to infect anything. They can infect us, they can infect plants, they can infect protists, fungi, they can also infect bacteria. And one of the main types of that one is a bacteriophage, and we'll talk a little bit about it later. These guys are characterized by a number of things though. So they are kind of put in groups based on a couple of things. First of all, what type of nucleic acid do they have? So when we look at a virus, they are going to contain either DNA or RNA. They're not going to have both. They're going to have one or the other. Now the DNA or RNA could be double-stranded or single-stranded. So this could be where they have a double-stranded DNA, they have a single-stranded DNA, they have a single-stranded RNA, or they have a double-stranded RNA. All right, but they only have one type. The second thing is they get characterized a lot of times by their size and their shape. Okay, so sometimes they get named based on the shape that they have, and of course then their size, because some are smaller than others. And last, do they have an envelope? Okay, so just like an envelope works for a letter and how it protects it, keeps it hidden, envelopes can also do that for viruses. So do they have one or do they not? So when we look at this, viruses are very diverse in their structure. They have lots of different shapes that could be out there. Again, different combinations of RNA and DNA. But there are two things that they all have, okay? So even though they can be very diverse, there's two things that all viruses do have. One is they do have an outer protein capsid. This is the part on the outside that gives the virus its shape, all right? And so it is the capsid. The other thing is the inner nucleic core. So remember, we talked about this is either DNA or RNA. So they do have a nucleic acid that's inside of the capsid. Something that's optional, and it is found mostly in animal viruses, is going to be if an envelope is present. But again, this is not something that has to be there, but some viruses do have it. So let's take a quick look at a picture here that shows you some of the diversity that we can see in viruses. So if you look here, we do have the first one is the tobacco mosaic virus. This is a virus that attacks tobacco plants. You can see it has a kind of rod shape to it with the nucleic acid on the inside, and it is an RNA type of virus. You then have adenovirus. Adenovirus is going to have this kind of multi-shaped isohedral type of capsid, and it's going to have a DNA nucleic acid. Adenovirus is going to attack into the nose. A lot of times it causes a cold, that sort of thing. We do see influenza. Influenza is the flu virus. This particular one has the capsids on the inside on the actual RNA, and it does have an envelope. So you can see that all of those capsids are inside of a special structure with the envelope. The last guy over here, that to me looks like if you've seen Toy Story, the original one, and the toys come out from underneath Sid's bed, the one that has the doll head on top of the little legs. This is what this one kind of looks like to me. This is a bacteriophage. This is one who attacks bacteria. So it's not going to attack us or plants or fungus. This one's going to attack bacteria. And if you look, it has DNA inside of it, and it has a very kind of unique structure to it. Kind of looks a little scary. So this is showing you some of the diversity that you can see. Now, when we look at this and we're looking at particular viruses, they are going to have a specific host range. Some of them are so specific that they only infect one thing. Others may have a range, okay, where they might be able to jump from one type of animal to another type of animal, okay, but they normally stay within a certain like kingdom or group. So the first one are the bacterial viruses. Those are known as bacteriophages or phages for short. Again, this is what that structure looks like, where it has the capsid head with the DNA. It then has this unique structure that injects the DNA from the virus into the bacteria. But this one is a very unique one. We also have lots of different plant viruses that can attack the plants. We saw the tobacco mosaic virus, and with that, it does affect us indirectly. Because if viruses start to infect plants, we see that our crops are going to be affected. It could be that trees are affected. There could be a lot of stuff that goes on with those plant viruses. Animal viruses, again, they can go from maybe one animal to the next. We see that the swine flu was able to jump from pigs to humans. Avian flu does not have the ability to do that as easily. It can happen, but it's pretty rare for that to occur. But when we do see animal viruses tend to have the envelopes present. Those envelopes help protect the viruses, specifically from our immune system, which we're going to talk about in part 2. So viruses, guys, are not classified with the normal Linnaean system of classification, like the taxonomy that we talked about back in the evolution type of chapter. They do have the capacity to mutate or change over time. The DNA can change, and this is what can make viruses where we have to kind of guess what we're kind of dealing with, like with the flu when we create flu shots. But that's also why we have different types like flu A and flu B, because mutations have occurred. They are also able to replicate or multiply, but they have to do it inside of a cell. They can't do it on their own. And so because of this, they are considered obligate intracellular parasites. So obligate means they have to. Intracellular means they have to get into a cell, and they are a parasite. They take over the cell, and the cell now just makes viruses. The cell becomes a factory of making more of the virus. Okay, and so that's how that works for their replication, or what we would consider them being able to multiply. Okay, now with this, they do take over the host machinery of like their ribosomes and any of the structures like the ATP and mitochondria and all the stuff they need in order to make more of those viruses. So let's talk a little bit about the replication process, because there's actually kind of two different pathways that viruses can use to replicate or multiply. So remember, this multiplication has to take place inside of a cell. And we see that we can have the lytic cycle or what we call the lysogenic cycle. Now, the lytic cycle is a shorter cycle, and you're going to see that it's missing one of the steps that you see in the lysogenic. So we do see that there is attachment, penetration, biosynthesis, maturation, and release. Okay, well, what does this mean? First of all, the virus has to attach to its host cell. So whatever cell it's going to invade, it has to attach to it. Once it is attached, it has to penetrate the outer cell wall or the outer membrane, depending on what kind of cell it is. And it has to inject its DNA or RNA into the host cell. That's the penetration. Once it gets inside, it does biosynthesis. This is where it hijacks the machinery in the cell and it makes more viruses. Biosynthesis, making more of it. The next step is maturation. And in this step, we're taking all the pieces that were made, the capsid, the nucleic acid, if an envelope needs to be there, we're starting to put all the viruses together. And then we're going to release them. Now, in this particular cycle, the lytic cycle, the release is going to actually kill the cell. The cells will die as they release more viruses into the host. On the other hand, the lysogenic cycle is longer, and it can have what we call a dormancy or latent period, where you don't actually see the virus actively replicating, but it's still present, it's still there. In this particular case, all the steps are the same as what you saw with the lytic cycle, but there's one added in. So we do see attachment. It has to attach first to the cell. We see penetration. We've got to get the DNA or RNA into the cell. Then we have something though that's called integration. Integration is where it takes whatever nucleic acid that we have and it converts it to DNA. So if it's already DNA, it can do this. But if it's not, it's going to convert it from RNA to DNA, and we're going to add it in to the host cell's DNA. And we call this a prophage. Okay, this is where it can sit dormant, it can sit latent, and we see that that particular cell could be infected for months or years before we actually see more viruses being made. Okay, and so it integrates in, and every time that cell goes through the process of mitosis and divides, it will copy this DNA as well. And so instead of having one cell releasing 10 of the viruses all at once, you may have, because it's been sitting dormant, you may have 10 cells all releasing 10 viruses. That is going to be a bigger infection load that can happen quicker, right? And so this one can cause some major issues. We do see that this is going to allow, again, for biosynthesis. Once a trigger has happened, it comes out of dormancy, and we see that it's going to make more of the viruses. It then will cause them to mature, and they will release them. Now, the release could cause the death of the cell, okay, just like the lytic cycle. But sometimes it doesn't. Sometimes it's going to bud out like through exocytosis. Okay, when we look at exocytosis, it means that the cell sends out something. This is where it might send out more viruses because it's been hijacked, and the cell doesn't die in this process. Most of the time, this is going to be for your ones who have an envelope, because what they do is they use your own plasma membrane or the cell's plasma membrane to be the envelope. This is why it can evade your immune system, which we're going to talk about a little later. So when we look at this, guys, the attachment a lot of times is a lock and key type of attachment. This is what causes them to be specific, where viruses are only going to affect certain types of cells or certain kinds of organisms, because you have to have the receptor in order to actually have attachment of the virus. When we look at penetration, the only thing that goes into the cell is the DNA or RNA. The capsid, the envelope, they don't get access. Only the DNA or RNA does. Okay, integration is the step that is different between the lytic cycle and the lysogenic cycle. So viruses are not actively being replicated, they're just integrated into the DNA. And then, of course, release is lysis or death of the host cell. That can occur, okay, in the release that's there. So let's take a look at this. This is the lytic cycle. So you'll see here first you have attachment. When attachment occurs, it's going to cause penetration where it puts in the DNA or the RNA. Now they're going to make more pieces of the viruses. They're going to assemble it and then release it. This is showing you the lytic cycle. So we're going to let it repeat. We have attachment, penetration, biosynthesis, we're making the parts. We're also going to see that it's maturation and then release. All right, and so this results in the host cell. See, the host cell is gone, it's dead. And we do see that virulent viruses, those that replicate very quickly and they can cause major damage to the host, are going to only use the lytic cycle. They are not going to get into that dormant type of stage that we see with the lysogenic. On our next one, we have the lysogenic cycle. In this one, we see that the first steps are just like the lytic cycle. So we have attachment, we have penetration. Then we see, though, that it's going to add to the DNA. So you can see that the DNA of the cell is green, but the DNA from the actual virus is red. But we see it gets added to that green circular DNA, and it can sit there. And as the cell divides, that viral DNA gets replicated as well. So the nucleic acid joins the cell's DNA. Viral DNA becomes part of this. That's what we call it as a prophage. This could go on for years before it actually triggers in order to make more viruses. Temperate viruses are capable of using the lytic or the lysogenic cycle, and these are things like HIV. This is why if you've been exposed to HIV, you don't necessarily test positive right away, okay? You don't have a high viral load that could be detected by a test. So you get tested right away, but then you get tested at 3 months, 6 months, 9 months, a year. If you are normally HIV negative at a year, you're probably HIV negative. We see cold sores. Same way, a lot of us have cold sores, which is a herpes virus. It's in your cells, but it's part of the DNA now, and it just sits there until you get stressed or your immunity is down, and then you get the cold sore. Shingles is also the same way. This has to do with the chickenpox virus, but again, it's going to be opportunistic. When it has the opportunity due to your immunity being low, whether it's due to stress or you're fighting something else, you might have a breakout of shingles. So let's kind of take a closer look at the two cycles kind of put together. So if you look here, you can see that the lytic cycle is that bigger circle, and it'll show you where we have, of course, the attachment, the penetration. We see that it's going through the process of making more of itself and that sort of thing. But the lysogenic cycle adds the part where we add to the DNA. This allows it to replicate multiple times. It sits dormant, but it will then eventually enter into the lytic cycle. Okay, and so this is what we're looking at for the two cycles. Now, the replication process within animal cells, like so viruses that have an envelope coat, there's going to be a big difference between DNA-based viruses and RNA-based viruses, okay? When we look at DNA-based viruses, you see attachment, penetration, biosynthesis, maturation, and release. That happens more often. But if it's an RNA virus, we do see that it's going to be more of that lysogenic side where integration is going to be more of an issue. Okay, so when we look at the RNA, they're similar to the lysogenic cycle, and HIV is a great example of this because it's known as a retrovirus. This is because it has the RNA and it has to go backwards and make DNA that gets added into your DNA sequence. Okay, retro means like old or backwards. It's in the past. Okay, and so that's where it's using that. And they package their own enzymes to be able to do this. We do see that penetration does have to occur in order to remove the coat. The nucleic acid and the protein capsid may enter into the cell, and this is something that could occur because they use that envelope. And this is because the cell could take in the virus by what we consider endocytosis, okay, where fusion of the envelope happens with the plasma membrane because the structures would be similar. Okay. So when we see that for release is going to be a little different where the cell may not die in the process. And this is because it's going to have that budding process, that exocytosis, where it's going to end up having the envelope coat to it. All right, so the last little bit I want to kind of talk to you about are examples of diseases. These are viral infections that are pretty common in humans. And so it shows you based on kind of the area of the body that is affected and some of the viruses that are there. Now, some of them are not as common. I mean, encephalitis and meningitis don't happen as often, but we do have some viral forms. Measles is a big deal right now. We do see there's an outbreak that's really close to us due to the unvaccinated that are there because herd effect is not taking place. and it can cause complications of meningitis, inflammation of the meninges and the brain itself. And so those are viruses like rabies, arbovirus, things like that. We do see the different types of common colds, which are like rhinovirus, parainfluenza, RSV. We see that you can have different eye infections like herpes, cytomegalovirus. So there's a lot of different versions. You can also see that some of these may start up in the head and then move down to other areas. So like influenza A and B, of course, are going to start up as like a head cold, but then they can go down into the chest and cause pneumonia if it's left untreated. You can see the different types of hepatitis, which attacks the liver, types of skin infections, sexually transmitted diseases. These just give you some of the examples of viral infections. All right, so let's talk real quick about treatment and prevention of these viral infections. So with treatment, we would use something called antivirals. We cannot use antibiotics when we're talking about viral infections because they won't work. Okay, so with this, antivirals are very limited. What they do is they try to support your body so that your immune system can then take over and get a handle on the virus. We do see that there are some that have been developed to treat herpes. One of those is acyclovir. It can help with keeping the virus from replicating. We see influenza. The flu, you've probably maybe had Tamiflu before when you've tested positive. And then we do have lots of different medications out there that they do in different combinations for individuals who have tested positive for HIV. We do have the most success with antivirals against something like HIV. There's a lot of research that's gone into this, but it's not really a cure. It's just to help keep the virus from replicating, making the patient sicker. Another thing that we can do with some viruses is prevention. Prevention is going to look at things like vaccines. Now, I know with vaccines, there's a lot of controversy about them, but there are some vaccines that have been around for a really long time that do really good things. And so it helps keep diseases at bay. Some people will say, well, those diseases haven't been around for a long time because of the vaccines. If you remove the vaccines, you are going to see it come back just like we're seeing with the measles right now. All right. And so we haven't seen cases of the measles like this before in a very long time, but now we are because people are not vaccinating their children. Now, if you have a religious reason to not vaccinate, that's different. Or if your child has an allergy based on how the vaccine is made, because some vaccines are made using eggs. If a child has an egg allergy, you don't want to give it to them. All right. And so some kids can't be vaccinated, but the other kids who can create this herd effect that can offer protection. So they may be prepared using a weakened live virus. In that case, we do see sometimes with the flu shot, that's what they do. Other times it's a killed virus where it just gives the proteins. The whole point, however, of a vaccine is for the vaccine to elicit an immune response so that your immune system actively works to fight the invader, okay, whether it's dead, weakened, that sort of thing. And it creates memory cells. That's the key. Because if you create memory cells, then the memory cells are going to remember that particular invader. So when you are exposed to it again, you have immunity already set up. You have immunity ready to go. Because the first time you're exposed, it takes a while for your immune system to come up with a plan and to fight it. But once it already has the plan, it can execute it very quickly. You may only feel bad for just a very short amount of time where you're like, man, I think I'm getting sick. The next day you wake up and you feel okay. Your immune system was able to take care of it. Okay, so live viruses lead to better immunity because it's going to give you a stronger immune response, but they can also make you feel bad and kind of feel sick. We do see that some vaccines are in continuous development due to high mutation rates. So this is one reason why it's called the flu shot and not the flu vaccine, because the flu shot you have to get yearly, and they're guessing which one's going to be the problem when they make that. Sometimes they're correct and it's like 80 to 90% effective. Other times they're not and it's only 15% effective. But regardless, they try with that kind of stuff. Another thing that helps with prevention is public awareness. When we do have outbreaks or when we do see that there's viral infections that are present, letting the public know about it and how we can work on kind of controlling it and keeping it at bay is really important. One of the main things is hand washing. Okay, another one is covering your mouth whenever you cough or sneeze. Used to, we would tell people to cover their mouth and they'd use their hand. The problem is then they do that and then they touch surfaces. Other people then touch surfaces. We teach kids nowadays if you'll use your elbow, it's a better version. Okay, but washing our hands is also a super important part with this. And with viruses, something else with prevention is vector control. We do see that a lot of viruses are carried by insects, specifically like different types of flies that bite or mosquitoes or things like that. And so vector control can be a really big help in keeping those viruses at bay. If we can keep the mosquito population in check, you won't see as many of those viral infections, okay? Vector control for like rabies is foxes and raccoons and things like that. Maybe even dogs. If a dog has rabies, we have to take care of that. And that's that vector control side. All right, so this finishes up part 1 of our kind of disease and immunity set of notes. If you have any questions, please let me know.